Metabolism is the sum of all chemical reactions in your cells. Essentially, the process of storing and releasing energy, but also the transformation of any given chemical to any other chemical inside your cells at any given time. It's a complicated topic, to say the least. One of the most important things that you have to understand before we can move forward talking about metabolism is the role of enzymes. Enzymes are a kind of protein. Way back in Unit 1 when we were talking about the biological macromolecules, we talked about proteins, and enzymes were just one category of protein. Actually, you know what? Hold on, I'm just going to bring up that clip of me talking about enzymes. This is something I can't do in a live lecture. Hold on. And then we have a big category of proteins, and this is enzymes. And you probably heard of enzymes before. Enzymes are a type of protein. You've heard me say several times now that your cells require a lot of chemistry in order to maintain homeostasis, in order to be able to do the work of being a cell, in order to do the work of being a living thing. Well, that chemistry can't really operate at the speed that is required by living things without enzymes to help them along. So enzymes are what we call catalyst. A catalyst is a material that speeds up a chemical reaction without actually participating in the chemical reaction. If you add an enzyme to a chemical reaction, it can happen 1,000 times or 10,000 times faster than it otherwise would have. And because the enzyme isn't used up in that chemical reaction, it can be reused over and over and over again. So just a small amount of an enzyme added into a particular reaction is going to have a big effect on the rate of that chemical reaction. Earlier, we looked at some things like dehydration synthesis reactions, right? When you put monomers together to make a polymer, like proteins themselves, and hydrolysis reactions, where you break down polymers into smaller units. Both of those are types of chemical reactions you get in the cell, but they would occur profoundly slowly if it weren't for the enzymes that are also present helping those reactions along. So here I have a man who is enjoying himself a nice roll, a nice bit of bread, and I've mentioned before that bread is starchy. It's got a lot of complex carbohydrates in it, so he needs to start digesting that starch. There is an enzyme in his saliva which we call amylase, and what amylase is really good at is finding those starch molecules and beginning the process of breaking them down immediately. So the digestive process, breaking down molecules, actually begins the second food gets into your mouth. So enzymes, they are biological catalysts, they speed up chemical reactions, and they're a kind of protein. Sorry if that felt like review for any of you, but I wanted to make sure that that information was fresh in your mind before we jumped into this material now. So whenever you have a chemical reaction, whatever you start with is called your reactant, and then whatever it becomes is called the product. But when an enzyme is involved, the reactant is called the substrate. The enzyme is going to work on a substrate, and then it kind of spits out the product. And generally, you don't have just one enzyme involved in this process. What's going to happen is you might go from substrate to product, but that product might actually become the substrate for a second enzyme to come in and then create another product. But that product might be the substrate for a third enzyme to come in, and you get what's called a metabolic pathway, a sequence of chemical reactions carried out by several different enzymes. Products from each step become reactants for the next, and in this way, energy can be stored or released during these reactions, and you can slowly and gradually change molecules and get very complicated as you move along. So you can take a really big molecule like a protein, and you can break it down into its component pieces, change things around, make alterations. You can take something very simple like carbon dioxide and gradually build it up into something much more complex. You could build it into starch molecules at the end of the metabolic process that you're working on. And metabolic pathways don't need to be linear either. They don't need to be a straight line. They can branch at a certain point. You can split your product into two different products, and then different enzymes can work on each one of those and take it in different directions. So you can have branching metabolic pathways. You can also have cycles where a substrate enters and it's changed in a whole bunch of different processes But eventually it circles back around and it gets added again to the original substrate, and the cycle continues round and round and round. But since we keep adding more and more of that substrate in, a byproduct has to fall out of the system in order to make sure that we're not just, creating more and more and more stuff. So you get a chemical cycle going around, a metabolic cycle, with a byproduct that leaves periodically. And when we talk about cellular respiration and photosynthesis, we'll see an example of a cycle in both of those processes. One is called the Calvin cycle, that's in photosynthesis. The other is called the Krebs cycle, and that's in cellular respiration. Now, in that clip from the long ago, I mentioned that enzymes speed up chemical reactions. How do they do that exactly? Well, remember when I said that sometimes in order to get a metabolic process going, you need a little bit of kickstart energy, right? You need a little bit of something to get it going in the first place. You need that activation energy, that energy of activation. Well, what enzymes do is they lower that requirement. They lower the activation energy of spontaneous reactions. And since it takes less oomph in order to get that reaction going, that means it's going to happen much, much more often, and therefore the overall reaction is going to happen much more quickly. Now, importantly, enzymes cannot make the impossible possible. They can only speed up what would have happened anyway. There's an enzyme called catalyze, which breaks down hydrogen peroxide. If you take hydrogen peroxide, you put it in a jar, just open on a counter, hydrogen peroxide will break down slowly over time, but it might take years, frankly, for hydrogen peroxide to break down on its own. But if you put a little bit of catalase in there, it can happen in hours instead. It'll happen thousands of times faster than it otherwise would have. And that's important because often the speed of chemistry that would just happen at room temperature or even at body temperature is not sufficient to maintain a living organism. You need enzymes in order to have living things with as much complexity as we are accustomed to. So in this picture here, I have a little segment of a starch molecule. You can see that you have some glucose monomers and they're linked together by bonds, and you got some water molecules around here. And of course you break down starch by hydrolysis reaction, right? So you can let water attack these bonds in between these individual glucose molecules, and that can happen naturally. But this enzyme here, which I'm going to say is amylase, although I don't think that's actually what this enzyme is, but come along with me on this. This is going to be an amylase enzyme, is going to hold the starch molecule, which is my substrate in this reaction. It's going to hold it just so that it's going to make it more likely for that water to attack it at the right angle. And because it's more likely for the water and the starch to hit each other just right, it makes it more likely for that hydrolysis reaction to occur. The energy of activation is lower as a result, and that speeds up that hydrolysis reaction. So when the starch substrate and the enzyme amylase come together, they form what we call the enzyme substrate complex, and then you get that transition form coming in with the water, the starch, the enzyme all together. The starch gets broken down into glucose, but you notice at the end, the enzyme isn't actually altered by this reaction at all. Amylase, it can just go on to the next starch molecule and can be reused over and over and over again. So this is why a single enzyme can speed up a reaction a whole bunch, because it's not consumed by the reaction. It can just keep going to the next molecule and the next molecule and the next molecule and the next molecule and the next molecule over and over and over again. You get a lot of utility out of a single enzyme. This next slide shows the enzyme as a kind of, it almost looks like a little landscape here. There's a portion of the enzyme that is called the active site, and the active site is where the enzyme connects to the substrate. The active site is perfectly fitted, it's perfectly shaped like a glove to fit onto the substrate. Enzymes are specific, they are hyper-specific to the substrate that they are meant to break down. So amylase is starch specific. Amylase will not work on cellulose, for example. It won't work on other polysaccharides. It is starch specific. Its active site will fit perfectly around starch, but not other molecules, right? So when the substrate bumps into the active site, it's like a key fitting into a lock. They tuck into each other just right. And the reason why they tuck into each other just right is because of the order of amino acids in the polypeptide chain and the way that they fold together. So again, the shape of the protein is a really big deal. And where did the order of amino acids come from? The DNA. So the DNA is a really big deal as well. And then with this binding, you can see up on this chart that the activation energy without the enzyme is really, really big, but with it, it's actually pretty small. It might be so small that the amount of energy that we just have in the environment is enough to overcome it, so we can break down this substrate into the products. We can break it down into glucose molecules, for example, and be able to use it for the next thing. Now, in the case of hydrolytic enzymes, where the enzyme is breaking down material, I tend to think of the active site as being the mouth of the enzyme. I think of it as going up to the substrate and biting the substrate, holding it in its teeth, and then like water molecules can come in and help break it apart. I tend to anthropomorphize things inside cells, which maybe is good and maybe is bad, but that's how I tend to think about it. But if you would rather the key in the lock analogy, that is very common. That's probably one of the most common analogies that's used for these things as well. So enzymes are specific to their substrates due to the 3D shape of their active site, which recognizes and binds to another molecule, the substrate, and enables the protein to carry out its specific function. And that function could be a lot of things. It could be just altering the chemical groups on the substrate. It could be breaking down the substrate, cleaving off groups. It could be binding things together. Not all enzymes break things down. Some of them build things up. It could be combining two substrates into a larger product, so adding them together. So enzymes can catalyze all sorts of different chemical reactions inside the cell. Multiple substrates can actually enter the active site. Sometimes you need more than one substrate in order to fill that pocket, and it will be precisely shaped in order to accept those substrates. And they will be held in the active site by weak interactions, hydrogen bonds, that sort of thing. Now, sometimes in order to bind correctly, the enzyme might need a little bit of help. It'll need something called a cofactor. This might be something like a mineral, something like iron or magnesium or something like that just helps it bind a little bit. It might help it take the right shape, or it might have the right charge that it would require in order to bind to the substrate. For example, some proteins need a little bit of magnesium, which is positively charged, in order to bind to DNA, which has an overall negative charge. That's one thing you would have called a cofactor. Other things are called coenzymes, which help bring some materials which are required for the reaction. ATP is an example of a coenzyme. The active site can lower the activation energy and speed up the reaction, which then is catalyzed by this enzyme. So the substrates are converted into products. Those products are released as new types of chemicals. So the chemical reaction has taken place, but then that opens up this active site to accept new substrate molecules. So this enzyme is allowed to be used over and over and over again. Just to drive this point home, enzymes are specific. The active site has to precisely match the shape of the substrate in a three-dimensional way. And if you recall, proteins are folded polypeptides. And that folding pattern is held together by hydrogen bonds. So the incorrect conditions, say it's too hot, it's too cold, it's too acidic, it's too salty, that can mess with that hydrogen bonding, which then is going to cause the protein to become misshapen, and the active site is no longer going to bind properly. We say that the protein has become denatured at that point. So this is one of the reasons why body temperature is something that's very, very important to maintain inside of humans. If your temperature goes way too high or way too low, all of your enzymes are going to be slightly incorrectly shaped, and they're no longer going to be able to catalyze all the chemical reactions inside your cells quite so well, and processes are going to start shutting down, and that's going to be a problem, a system-wide kind of problem for you. This is why the pH in your blood is maintained with a buffer system, and why the enzymes in your stomach are built to operate at low pHs, whereas the enzymes elsewhere operate at different pHs. So that is a term I'll probably test you on. To denature proteins, to denature means that it has lost its shape, and when it loses its shape, it can no longer function. So you see a nice folded polypeptide up here with its active site in red, but when it becomes unfolded, all the amino acids that were part of the active site, look, they're all over the place now. It's denatured, no active site, no binding, no catalyzing reactions. Doesn't work anymore, non-functional enzyme. And let's talk about those coenzymes and cofactors. Coenzymes are organic, that's a key term there, organic carrier molecules that deliver materials for reactions. For example, in cellular respiration, we will talk about something called coenzyme A, which delivers acetyl groups for the reaction. We will also talk about something called NADH, which has the job of delivering high-energy electrons. And also many vitamins that you require in your diet are coenzymes, or they will be derived into coenzymes later on. ATP is a coenzyme. Cofactors are usually inorganic materials needed for the enzyme to help bind to the substrate. This includes many minerals required in your diet, zinc and iron and magnesium and these sorts of things. And I already told you that magnesium is a good example, helps bind DNA polymerase to your DNA because DNA is negatively charged and magnesium's positively charged. DNA polymerase is an enzyme that we will talk about in Unit 4 when we're talking about DNA replication. There are two broad categories of metabolic reactions I want to talk about. So metabolism is the sum of all chemical reactions inside of your cells, but then we can divide that into two subcategories. There are the anabolic reactions and the catabolic reactions. Anabolic reactions are the ones that build up molecules. We start with small starting molecules and we put them together, we link them together, and we build up and up and up and make bigger and bigger things. And typically these are energy storing reactions. You've heard that term anabolic before, right? Anabolism, anabolic steroids, right? What do? You get some, I don't know how it works entirely. You inject something and you build your muscles up, right? Bigger. Well, that's the same way this term is being used. You're building things up. You're increasing size. So you can use that as a mnemonic, if nothing else. So anabolism is to build things up. A good example of an anabolic energy-storing, bond-forming reaction is photosynthesis. This builds up big molecules from small ones. Every enzyme in this process is catalyzing a reaction wherein energy is being put into the system. So we're starting with small starting components, getting bigger and bigger, more and more energy adding to the system. You can think of anabolic reactions as putting bricks in to build up a skyscraper. So we're starting really small, but we're going to get more energy into the system. Think about how much energy is required to fuel these construction machines. To put in these bricks, right? Every brick that you put up, you're lifting it up higher and higher and higher. You're putting tons of potential energy in there. You have to feed your workers in order to get them to do their job every day. Lots of energy required to build things up, and time. Oh my God, so much time and energy to build these things up. Dehydration reactions are a good example of anabolic reactions. Dehydration reactions, you remember that from Unit 1. This is how we build polymers. You take two monomers, dehydration reaction, you end up with a dimer. Another dehydration reaction, now we're starting a polymer chain, right? Another dehydration reaction, we make it a little bit longer and longer and longer. Opposite of an anabolic reaction is a catabolic reaction. When you hear catabolic, think catastrophe, right? We're breaking things down, we're destroying things. We start with a big molecule and we break it down into smaller pieces. Big molecules have a lot of stored energy in them. They are highly ordered states. They are in low entropy, and we're going to break them down into smaller units. We're going to break them down into high entropy states. We're going to release energy from them in catabolic reactions. The most important energy harvesting bond breaking reaction is cellular respiration. And another good example is hydrolysis reactions, right? Which is the opposite of dehydration reactions. So in this metabolic pathway, we start out with something pretty complicated, and every enzyme along the way is going to catalyze reactions which take a little bit more energy out, and we're going to get smaller and smaller as we go. So if building up a building is a good example of anabolism, then you can think of demolition as a good example of catabolism, or a good metaphor for catabolism anyway. Think about how fast demolition is compared to construction, right? You just gotta set a few charges, have a little activation energy, if you will, and then the whole thing comes tumbling down. Why is it so much faster to knock things down than to build it up? Because all the potential energy is already stored in the system. All you're doing is releasing it. All you're doing is giving it a little bit of a kick, a little bit of activation energy, and it all comes tumbling down. So it's much faster. That way, it's an energy releasing reaction. So catabolic reactions start with big molecules, break it down into small ones, and release energy along the way, and that's cellular respiration. Now, before we can get to photosynthesis and then cellular respiration, we have one more bugbear to slay, and that is redox reactions, which will be the topic of the next video.